Well, thanks, Jen. And yes, I'm going to talk about how to be the perfect host. But I thought, first of all, since the BRC are hosting this, I just give a little bit of an idea about what's going on in the Biomedical Research Centre in terms of immunity. So immunity is in many ways relates to many aspects of medicine, as well as being a sort of speciality on its own. We have set up what's called a core lab. It's the scale is it's actually a relatively small core lab, which does some of the assays which I'm going to describe a little bit about the basis of in the talk. So try to use new techniques to analyse immune responses. But we've got links to clinical immunology, which look after patients who've got immunodeficiency, renal medicine, infectious diseases, which is really what I'm going to focus on today. But also when the immune system goes wrong and is overactive in terms of rheumatology and autoimmunity, dermatology and allergy, and also links but not an actual subtheme in transplantation. So really these are all areas which are in some ways clinically very diverse, but linked through this idea that there's some kind of common patterns of the ability of the host to deal with things that are thrown at it, whether they're viruses or transplants or bugs of other kinds. So this is going to be a very general talk about some principles that underlie both virology and immunology. And the first thing I'd like to get across is what is a virus. So there are lots of obviously misconceptions about what viruses are. So this one is a quote from Peter Medawar, who was a Nobel laureate who worked in Oxford, he had a very negative view about viruses. He called them a piece of bad news wrapped up in protein. Actually, viruses got more to offer, I think, than that. You can use viruses to deliver vaccines against all sorts of things, and you can use viruses as gene delivery vehicles, and they're being used to treat all sorts of, or potentially all sorts of very significant diseases. So although he was very upset about them, I think we should look a bit more on the positive side. But the fundamental point is that viruses grow inside body cells, and bacteria typically grow outside the body cells. So the sorts of host defence system that are set up to deal with bacteria and viruses are quite different. Some things that will deal very well with bacteria are not very good at dealing with viruses, and vice versa. So bacteria, here's pictures of bacteria. I just put my pointer on, see. Some bacteria growing on a plate. You can never grow viruses on a plate. You need a cell layer, and viruses really can only be seen using an electron microscope. So there's some. Should we put the lights down a little bit? You think it would probably be. It's just So my work's mainly on a virus called hepatitis C, but in terms of antiviral responses, here's a typical virus. So it's got an outer shell, and on the outer shell it will have an attachment protein, and those attachment. This is true for basically all viruses, and they'll have some kind of attachment protein. These are quite variable proteins generally, and they allow the virus to decide what animal it's going to infect, and when it gets into the animal, exactly what cell it's going to go into. And these, of course, are fantastic. You can imagine that's a fantastic target for the immune system, because if you can only attach an antibody to that protein, you might be able to block the entry, and that's been the basis of many vaccines in the past. And much of our initial understanding of how host defence worked. But actually, it's a bit more complicated than that. So inside a virus, a typical virus, you'll have some kind of inner packing protein, because the viruses are very geometrically arranged. So the inner proteins are all bound in a way that allows them to kind of get together like a load of Lego blocks and form a nice solid mass that will support the inside of the virus. Now that is an excellent target for the immune response, because unlike the attachment proteins, which can be quite variable and change their structure, the inner packing has to be, by nature of it, it's the fact that it has to pack in very tightly, it can't change very much. So the immune system is very interested in those. It's got to find a way, as I'll describe later, of actually accessing that little bit of the virus. And then you've got the viral blueprint, which can be DNA and can be RNA, and viruses use all sorts of bits of backwards and forwards DNA and RNA in order to replicate. And that's not very well recognised by the host immune system. But interestingly, just the presence of some of these things, not the specificity of which virus it is, but just the nature of having some viral RNA in the cells is actually a very good way of turning on your innate immune system, as I'll come to. Okay, so here's some pictures of some viruses. So here's a virus from the herpes group, and this is the core of the virus as I've described, and there's a kind of envelope around it. I think this one looks a bit like a bunny rabbit. And that's cytomegalovirus, which will affect about half the people in this room. You'll be fine with cytomegalovirus. In most cases, it doesn't really cause any harm, and as I described, we live very happily with that sort of virus, as we do with many viruses. This is hepatitis B virus. Hepatitis B is a major cause of hepatitis globally. About 300 million people have got hepatitis B. And if you add that to the 200 million people who've got hepatitis C, that's 1 in 12 people globally have got one of these viruses. So this is the virus itself. And all these sort of funny little blob things are aggregations of viral protein, and the virus actually makes these aggregates in order to basically put off the immune system. They're decoys, and with a few viruses in here. These were picked up by Baruch Blumberg, who won the Nobel Prize and was a former master of burial here in Oxford. So here, just two extremes, and in fact many people with hepatitis B virus will live with it quite happily, although it can cause very severe liver disease. So how do viruses grow in cells? Again, this is very simplified, but it's very important in trying to understand how the immune system can really make an impact on the viruses. So a virus will get in using its attachment protein. Again, it will depend on the cell and the virus and so forth, and it will uncoat, and then it's got to go through a clever little trick to get itself through that cell membrane, which different viruses solve in different ways, but it ends up within the cell, in the cell cytoplasm, and then it's got to engineer itself so that it can use the cell's machinery in order to multiply. And it harnesses, most viruses are not bringing in very much with them. In fact, my favourite virus is a virus called parvovirus, which has only got 2 genes. We've got thousands of genes to keep us going. This virus has just got 2 genes. One for the outside coat and another one which we don't even know what it does, but it still manages to infect about 80% of the world's population. And it does it in just the same way. It harnesses everything it can from the cell in order to multiply. So you'll get one piece of DNA, and from that you'll get very many viruses. And that's really the important point about this slide, is that there's a massive exponential growth curve to viruses. And the main aspect of the immune response is to try and get on top of that growth curve as early as possible. Because the same immune responses, if you bring them in later, there's very little they can do. All they can do is try and play catch-up with the virus. If you can get them in early, they can actually arrest the thing in the first place. And here's a picture of a virus doing just that. You can see it's all bursting out the cell. This is a beautiful picture. In fact, it's actually chilling, really. This is an HIV-infected cell. And here the virus is budding from the cell surface. The viruses come out of the cells in different ways, but HIV actually buds from the cell surface. And if you look carefully, you can sort of get the impression of little bits of virus under the cell surface here, which are just getting ready to go. So one cell can produce millions and millions of virions in a very short period of time. What's the immune system actually going to do about this? So how do we defend ourselves? So I'm going to use this is going to be an analogy which works for a bit, and then you'll have to just bear with me. So you've got to imagine that your body is like a stately home. In the UK, it could be in any country actually, but the stately home, this stately home is Blenheim Palace, as I'm sure you'll recognise. I think the John Radcliffe is somewhere in the background here. I like to imagine it's somewhere in the background there. But anyway, there's Blenheim, and essentially there's a great party going on at Blenheim, and the virus is not invited. Okay. But of course you're going to have lots of other guests at the party. So your job as the host immune system is not to defend everybody from getting into Blenheim. It's just to stop the ones you don't want. You don't want the gate crashers. But you're allowed to have other things in. So you eat lots of food. You're continuously exposed to bacteria every day on your skin, in your bowel. As I'm sure Derek Crook told you, 90% of the cells in our body are bacteria. So we're completely covered in foreign material, but we just don't want the viruses. We don't mind all those other ones. So this is a very accurate depiction of Blenheim Palace with the party, and you've got a security, which we'll come back to, and you've got a host defence, and viruses have got to get in, and they want to get into this main area, and around the palace, you've got a series of defence mechanisms. So first of all, you've got a moat, and we're all very keen in the Biomedical Research Centre and in the labs in Oxford in looking in real detail at the host immune responses, and we've got molecules and a very kind of detailed things that we look at. In fact, some of the most important things are the most simple, and the skin is a very important host defense against viruses, because viruses have got to grow in living cells. So viruses cannot grow in a dead skin cell. The outer layer of your skin is dead, so the skin acts as a wall of death. So viruses usually pick a much easier route, so they're going to avoid coming in through the skin. They'll pick the path, the main entrance, which will be coming in through your mouth with all the food and other things that are coming into the party, or the respiratory tract. Or sometimes, in the case of the medical profession, we parachute the viruses in by using an injection, and that's been responsible for spreading viruses like hepatitis B and C and HIV throughout the world. So that's one very, very simple thing. And then in the grounds of the castle, you've got dogs. And we ignored all this element of the immune system for a long time, but the dogs are playing an incredibly important role, as I'll come to. So dogs have a fantastic nose. They're very able to sense very small amounts of chemical in the air, and they've got fantastically sensitive ears and probably lots of other great sensors as well, but I'll stop at those two. And they've got two great things that they can do when they sense something. They can bark, and they can also bite. Okay. So the point is that you have an innate immune response, which is set up very similar to a dog. And the dog uses its sense of smell and its sense of hearing to sense danger. And so it doesn't necessarily need to know exactly what the source of the danger is. It just needs to know that there's something out there that shouldn't be coming in. It's a gatecrasher, but they don't know whether it's a gatecrasher from Bladen or a gatecrasher from Oxford. They just know that they oughtn't to be there. And the barking allows them to alert security, which will amplify the response. But additionally, they can see off people single-handedly. They don't necessarily need somebody else there. So, and the things that we consider are important, these are a group of chemicals called interferons, some of which we use in patients. And an important group of cells which have been recognised recently are dendritic cells, which collate these danger signals and then also act in some ways like the dogs. So in the same way that you'd set up your own senses to recognise danger, so you can hear me, obviously, and you can see me, and probably the people in the front row can smell me as well. You have lots of impressions of me, and that's so the same thing's true for the innate immune system. You don't want to have a system that could just rely on one particular sense, because you don't really know where the pathogen's coming from, and pathogens, as I'll come to, are very smart. So if they knew that you could only sense them by hearing them, they would just keep quiet. So you have to have a range of things to activate the innate immune system. So dogs are effective and very quick, but they're probably not smart. You don't want them running the whole show. You need a bit more than that. And the adaptive immune system is important in allowing you the specificity of getting rid of gatecrashers that you really don't want, and also distinguishing them from guests, so that the dogs might tend to kick out the guests as well if they were unleashed. But you have to have, at the same time, a specific immune response for a specific virus and also specific substrains of virus, and that takes time. So the innate immune response can come in very early and deal with those big bursts of viruses, and so the security that comes in later, the rest of the immune response, has got, in principle, less to do. So if we're talking about what these are, you've got white blood cells which are found in the blood and also in the lymph glands. So in fact the majority of them are not in the blood, they're in lymph nodes, they're in the spleen, and they're also in tissues like the liver and the lung. They're spread very widely. And we've got a series, and the colours are totally unrepresentative of what they really look like. They're actually not very coloured at all. But you've got hand-to-hand killers, which are the killer cells. You've got logistic experts, which are the helper cells, and then you've got cells which make antibody, which are the B cells. And those are the key ones that we focus on in terms of antiviral immunity and host defence. So if you think about what the killer cells can do, they have got a specific job, which is to get rid of viruses which have ended up in the room of this stately home. So the dogs will sort things out in the grounds, but if a gatecrasher gets through and gets into one of the rooms, the dogs cannot get in. All they can do is stand at the door and bark. So the killer T cells need to perform a room-to-room search, and they need to investigate rooms where you've got a specific virus, so that they don't want to, the vast number of rooms, obviously, and you've got a vast number of cells to survey. You only really want them to focus on the ones where there's a problem. And they need to clear out the room, ideally, by getting rid of the invader. But if they can't clean it out, they have to trash it. That you've got this bimodal action of the T cells. They're either going to clear the thing out and leave the room intact, or if they cannot do that, they have to destroy the room and obviously the virus with it. And then that leaves the antibodies to deal with anything that's not in the room and soak up the rest of the viruses which are in the corridors and the grounds. So that's a division of labour. So how do they do this? And this is a really sensitive system, and this was worked out about, well, it's probably about 300 yards away now at the other end of the corridor. Alan Townsend, who worked with Andrew McMichael, was really fundamental in trying to understand how the killers could envision what was really an infected cell, how they could tell what's an infected cell from an uninfected cell. And to cut many years of work short, the virus gets into cells, as we've already described, and then it replicates. And the proteins go through a natural process of degradation. So all of us, all the time, we have proteins that are being made in our cells and they're being degraded. There's a natural turnover of proteins. And the immune system harnesses this to present some of these proteins on the surface of the cells. So the cells have a special presentation molecule on them called the MHC molecules. And these present peptides, and these are bound by specific molecules on the surface of the killer cell. So this interaction is incredibly specific. So this killer cell will only recognise a particular viral peptide. But the target could be any cell. So all cells have got the capacity to do this. So it doesn't matter that if the virus gets in your big toe or your brain or wherever. There's always this potential for the killer cells to come along and survey what's happening. So the recognition molecule then triggers the killer, and it may kill the cell, and therefore, or release a number of factors which can kill the virus, and potentially it can kill the cell. But there's obviously a disadvantage, as I said. If it kills the cell completely, it will get rid of the virus, which is good, but you will also cause tissue damage. So the immune system is always trying to weigh up what the advantage is in killing a particular cell. It gets a disadvantage. If you have an immune response in your brain which gets rid of the virus in the brain but leaves you with a lot of brain damage, that's not advantageous. So over periods of evolution, the whole thing's been balanced out. But viruses have basically taken advantage of this balancing act and tried to exploit all the various elements. So here's a picture of a killer cell doing just this, and it's a video. And the cell on the right and on your left here is a killer cell. It's actually from me, and it's been grown in culture. And these cells are still growing, and the person who grew them long since left Oxford and has now got a lab elsewhere. And I went to visit, and they said, "Oh, you're the person with the cells," because the cells have been growing in culture for many years. And this is a tumour cell, which is fortunately not from me, but it's just a good target for these. And they've coated the tumour cell with viral peptides. And what you can see coming up is just the interaction. And you can see these little red dots. These are small granules which contain a lot of enzymes which the killer cell is going to use to destroy that target. So as you can see, the T cell's it's got a good grip on the target. It's formed what's called a synapse, which is a word that comes from neurology, where two neurons sit together. But there's a very, very tight interaction just around here. It's transmitted some of these red granules into the cell, and they delivered basically a kiss of death to the cell. And I like that one so much, I'm going to show it again. So you can see them get together, and this cell is shrinking massively. And what's happened is the granules have gone in, and the cell has undergone a process of apoptosis, or an automatic cell death. And this blebbing is disruption of the cell membrane. So you get the feeling that this cell is really going to die, and it can be quite quick, within a few minutes, that this thing can go on. And these killer cells can then go on to search the next room and the next room, and that way suppress the viral infection. So how does it work then over time? So if we go back to our analogy, so this is a specific set of T cells has been activated, and let's imagine the virus is in the west wing, so it'll be localized in, for example, the virus I'm interested in, the hepatitis C, is largely localized in the liver. So the virus will come in, it will replicate, and it will reach a certain magnitude. And the immune system is always, there's always a little bit behind. So there's got to be a certain amount of time for the immune system to build up the numbers of the T cells they need to proliferate. And you generate what I've called here a red squad, so a very specific set of cells for that virus, which will have existed in very, very small numbers beforehand. And they need to migrate from the lymph node, which is where the proliferation is taking place, and they receive signals which allow them to migrate, in this case, let's say, to the liver. So they'll go out there and they'll deliver those kisses of death to the virus-infected cells. They'll clear out the thing, and in an ideal situation, that would be the end of it. So the innate immune system would get on top of things early. They'd tell the adaptive immune system that there was a problem. The adaptive immune system localizes the infection and eliminates it. So that's the perfect outcome, and that happens with quite a few viruses. And over time, of course, we're all exposed to lots of different viruses, many of which we deal with very well. And so we have a set of T cells which are specific for a range of viruses, and that forms the basis of immunological memory and, of course, the basis for vaccination. And this is how we measure that now. And now these tetramers, this thing that here is called an MHC tetramer, and this is the modern method for counting all these individual immune responses. And this was actually developed locally in Oxford. And what we've got here is the presentation molecule, the MHC molecule, and a viral peptide sitting in the groove. So these are the things that sit on the surface of the cells and offer up the viral peptides to the T cells. And what we've done here is recreate them in a test tube using recombinant DNA technology. So we can make the ones we want. And then we fix them together in a group because we fix them around a molecule which is fluorescent. And that means when a T cell comes along and binds one of these, we can identify that T cell because it will light up in a laser. So this is blood from a healthy volunteer. In this case, it's not actually me, but it's a healthy lab volunteer. And we've taken their blood, and this could be it could be half of you in the audience, because the MHC molecule that we've used is a very common one. It's present in about half of the population. And most of you will have been exposed to, again, very common virus called Epstein-Barr virus, which can cause glandular fever, but is usually dealt with very well. And we've taken their blood and we've made some of these reagents, and we can now identify, by passing them in front of a laser on what's called a flow cytometer, the cells which are killer cells. So these express a particular marker called CD8. And so everything above this line here expresses CD8. So these are all the killer cells in your body, or all of the ones in that sample of blood, rather. And these ones are the ones that bind the tetramer. So quite a nice population of cells. You can see that each individual dot there is a different cell. There's a great big population. It might be several percent, often 5 or 10% of your CD8 T cells, your killer cells, are specific for just one portion of Epstein-Barr virus. So some of these memory populations can be really quite large. And the reason that this one's particularly large is Epstein-Barr virus tends to hang around a long time, so it's got quite a long time to boost up that squad. So that's how we count the cells. And then if you do these, if you count the cells and count the virus, you get some idea of the dynamics of the response. So this is actually just a cartoon of what might be happening. So if you use look at an infection, for the first 5 days or so, you actually don't get that much of a T cell response. As I said, you need a lag to build up these T cells. And when the T cells come along, and they often peak after a week or so, but in the case of Hep C, it can take many weeks for this to build up. And then, once you've got the CD8 cells that are able to kill the virus, you can see the virus load will come down to some very, very low level. And then you get long-term control, and you have these T cell responses which last for a long time. So you have a long-lived memory. So that's how we deal with viruses, and on a good day. And how do viruses deal with us? Because then basically viruses are much smarter than we are, and they've had a long time to figure out how our immune response works, and therefore how to get around it. So it's not really a fair challenge, actually. So we've resisted over time mechanisms we've developed evolved mechanisms to resist, and viruses have evolved mechanisms to survive, but they do it much quicker than we do. And essentially they fall into two camps, broadly. Viruses which run away from the immune system by being quicker than we are, and viruses which hide from the immune system. And I'll give you one example of each. So but often, as in most simplifications, the viruses will use more than one method. And HIV is a particularly difficult one to get rid of because it does everything. It hides away and it runs away, and it's just a really difficult one. Hep C is perhaps slightly easier, and that's why I think we've got a better chance with the vaccine, as I'll show you at the end. But I'll start with cytomegalovirus. As I've already mentioned, it's a virus which many of us have been infected with. In fact, globally, about 80% of the world have been infected with CMV, a bit like parvovirus. But most of us, I'd be surprised if any of you knew that you've been infected with cytomegalovirus. It's very rare that it causes an acute problem, but it can cause a problem in people whose immune system's not working. So normally you get infected from your mum, in fact. It's just one of those things you get from your mother. And an infection is normally by the oral routes, but it's in breast milk, and there's a lot of early infection. And it will replicate in your local lymph nodes and glands, and then it goes into many places around the body, so it'll spread around. Again, as I said, not in a very clinically significant way. And then it disappears from view, and you'd be forgiven for thinking that it had disappeared entirely, but that's absolutely not the case. Because the price of this, of not being able to detect the CMV, is that you, as I've already kind of hinted at, is that you've got very, very large numbers of T cells and immune responses which are devoted purely to keeping the CMV suppressed. So this is what happens if you don't keep the CMV suppressed. This is the eye of a patient who's been highly immunosuppressed, and the CMV in this case has come back in the eye. So, and this can happen quite quickly when you're immune suppressed. And so it's quite a chilling thought, because what's actually happening in all of us who are infected with CMV is we have a very active immune response, and that's the difference between having a normal eye and an eye that's full of virus like this one. And it's a very finely tuned system that's keeping us well from these viruses. But the viruses have also evolved over time, so that they're relatively well adapted to the host. And it's actually, CMVs are very interesting because they co-evolve not just with us, but they co-evolve with all sorts of different animals. So rats have got their own CMV, and mice, and in fact, even looking back to invertebrates, some of these have got CMV. So these are very, very clever and ancient viruses, but they do demand a lot of attention, so you can't turn your back on them. So what happens in CMV? So again, it's the same as before. The virus comes in. It's not totally localized, but let's call it localized in the west wing to start off with. You build up your immune system, and it comes in, and it looks like the virus is going away, but it hasn't, because it's just going to pop up elsewhere. And if you do very, very detailed studies, you can identify very, very small foci of virus that are just popping up at very, very low levels and then disappearing again when the immune response comes chasing after it. And what the virus is actually doing is messing around with the really fundamental elements of the immune presentation. So the virus comes in and replicates, as we discussed, makes new proteins which come through this processing machinery, this sort of sausage machine which chops them up into peptides. And what's absolutely essential for the T cells is that the peptide is presented on the surface of the infected cell. And what the virus does is it produces little spoiler proteins which just get in the way of this process. They stop the peptide getting to the cell surface, and therefore there's no signal for the killer cell. So the virus has really exploited the weak links in this chain and produced a very specific protein which turns this thing off. So it's a better immunologist than anybody in the BRC, I'm afraid. And this is what happens as a result. So this is if we think back just a few slides, I was mentioning those tetramers, and you can use these tetramers in blood from lots of different patients. And if you just look at it by age, over time you get an increasing number of the responses, because each time that you can imagine your immune response is chasing around after the CMV, and it's getting bigger and bigger over time. So that we call this memory inflation. So there's a massive amount of your immune response is dedicated to looking after this one virus. That's just another picture of CMV. So that by the time you're 80 or so, actually about a third of your immune response is dedicated to this one particular thing. There are lots of other herpes viruses that hide. And it tends to be the bigger viruses, because the viruses need to encode specific information in order to do these clever tricks where they interfere with the presentation. So that involves usually having an extra gene. So you have to be quite a big virus to do this. If you're a small virus, you can't necessarily encode all this stuff. So Epstein-Barr virus and lots of other viruses in this so-called herpes family do it. And HIV is very good at doing that, and that can actually become truly latent. What about running strategies? So running is just a very simple thing. So the virus comes in to the west wing. It grows. The immune system is built up, and again comes to the right location as previously. But then the virus will change colour, and then the virus is visible in principle, but not to the red squad. So you've built up one set of immune cells, and in fact, if you think about it, you've effectively eliminated the red virus, but another version of the virus is still there. So you've now got another foe, and you have to build up a new immune response all over again. So you've lost all that time, and that can go on and on. And many of these viruses have got a fantastic capacity to replicate, and the concept is that each time it replicates, it makes, as I'll show you, can make a new virus, and all of these viruses are continuously present at any one time. So this virus has always got the capacity to be one step ahead. So if we think about hepatitis C, so somewhere around 170 to 200 million people have got hepatitis C globally. So it's a massive problem. If you think about the infection that it causes, this is just acute disease. Actually, it's very non-specific at first, and we don't catch many people in acute disease. So people will feel generally off colour. They might have some nausea and vomiting. Their liver might get a bit big. And then they might come to medical attention. People don't, all those things I've described, apart from the big liver, which you wouldn't necessarily notice, that could happen with almost anything, but people don't like going yellow, so they'll probably come to medical attention when they turn yellow. But lots of people don't even reach that severity of illness. And then for those of you who are really interested, you get pale stools and dark urine, which is a sort of end of the thing. But that's just acute disease, and it's unpleasant, but that's really not the problem. The problem is that hepatitis C tends to set up chronic disease, which is the majority of cases. So how's this running process work in hepatitis C then? So you start with, again, this sort of prototype red octagonal virus. And then this is a representation of its genome, which is relatively small. There's 10,000 base pairs in it, so that's tiny compared to ours. And then at one position, it might make a mutation, and that would change it from red to green. Another mutation, another position, might actually be deleterious for the virus. So obviously the virus is making mutations, some of which are good for it and some of which are bad for it. So there's a bit of a trade-off actually. If we made this many mutations in our cells, we'd have tremendous problems, because every time a cell replicated, it would have a potential defect in it, potential cancerous cell would form, and we have to maintain a much more stable genome than these viruses. But the viruses, they don't really care, they'll just make another virus. So if that one doesn't work, well, I'll make another one. So they can go through this process because they're replicating at such high levels. So the colour change leads to poor recognition by the ongoing immune response, and so that then replaces the red virus, and then the whole process can go on again. So there's really very little limit to the amount of essential genetic space that the virus can explore. So instead of just having a red strain which is eliminated by the red squad, you then get replacement. And you can actually view this in real time in patients by sequencing their viruses and getting a view of which virus is present at any one time. And you can see the disappearance of some strains, the re-emergence of others, and you can see the emergence of different kinds of immune response as the thing continues. And so forth. So this is really the running strategy. So as long as the virus can keep generating new variants, it's very difficult to pin down. So the result in Hep C is that 85% of those infected become chronic carriers. And once somebody's infected, it then becomes very difficult to eradicate it, because your game of catch-up is very hard, because the virus is a long way ahead. And in terms of vaccines, we've got issues here, because we need to figure out which of these multiple different kinds of virus we're really going to try and eliminate. But we're not daunted by this because, as I'm going to show in a minute, we're generating vaccines against hep C. So how do you improve security? So this is back to our Blenheim Palace thing. So some stuff you can't really do much about. And in fact, you could, in principle, have a system where you had more innate immunity and build up a very large squadron of dogs that just did a lot of barking and biting. But there is a risk in that because you put yourself in, if you think about the balance, you would be a bit high risk of autoimmunity. So that really the strategy is to build up more of the adaptive immune response. And back to the idea of the timing of it, you might not need that many security. You might not need a huge security operation which was active at all times, but you just need to get them out very early in the process of infection so that they can expand and knock the virus on the head at a very early stage before it goes through that exponential expansion. And how many do you need? Well, actually, this is probably a hard slide for you to see all the details of. But this is an experiment where we try to work out how many cells you have that are protective against influenza. So we know that if you've had influenza before and you see the same strain, you'll fight it off. And at the core of that, you've got a set of helper T cells which are instructing your immune response how to work. And we made, again, these tetramers to try and count these cells. And actually, unlike that slide that I showed you earlier, we had very, very large numbers of T cells against CMV. In fact, we worked out that on average you have 0.0061% of your T cells. So there's a really tiny population here that you can only see when we did kind of lots of extra enrichment to see this very small population of cells. So it's not many, but they respond very quickly. And so actually this was quite encouraging because flu is a good example of a virus which is complex, but nevertheless good vaccines are being generated. And so you don't have to have a vast number of T cells there, but you just need some primed and ready to act. So this is how we're doing it for hep C. And the point about hepatitis C is that the variation and all that variation from one colour to the next isn't uniform throughout the virus particularly. There's some regions which the virus has to keep more conserved than others. So it doesn't mind changing the external binding proteins, but it does have a big problem changing some of the internal proteins because those are involved in the packing and also the replication of the virus. So, but we do have to make a choice, and we've gone for one particular genotype of the virus, and this is probably a limitation, so we won't be able to cover all the different genotypes that cover the different parts of the world, of which there are 6. But we'll cover genotype 1, which is very important in the UK, and perhaps we need to make another virus vaccine for the next one, so we'll be busy for the next little bit. This is very conserved, and it has multiple epitopes. And in order to make it safe, this is done in collaboration with a European group, and that group have just inactivated the remnants of the virus. This is only a bit of the virus. So it's really can't form a proper infection, but just to be absolutely sure it can't do anything, it's been essentially paralysed. And this work's being done with my colleague Ellie Barnes, who's a hepatologist and has been really instrumental in getting this programme off the ground in Oxford. What we've done is, together with the vaccine theme and Adrian Hill, we've inserted that bit of virus, bit of hep C, in another virus. And this is an adenovirus. And this particular adenovirus came from a sick chimpanzee originally, which doesn't sound very good, but I can reassure you that it was harmless. It's been rendered harmless because it cannot replicate anymore. It's had all its insides taken out. But the reason it was taken from a chimpanzee is because humans will not have seen this virus before, so they won't have any prior immunity to it. So adenoviruses are really very good vaccine vectors because they can be disabled completely and made totally safe. You can put the foreign antigen, in this case hepatitis C, in, and most importantly they induce strong T-cell immunity. So all of those things make them really excellent, and we've just happened to have used one from a chimpanzee. We've got 3 studies going on, and I wouldn't worry too much about the details of these, but the first one is nearly complete now, and we've taken healthy volunteers and we've given them these viruses, these vaccines, in a graded way, and we're looking at safety just to reassure ourselves that many adenoviruses have been used previously, but we really needed to know that these ones were safe. and also that they generated T cell responses. And we're going on with Ellie Barnes and other colleagues to try and see whether these vaccines can also be used once the infection is established, to try and knock the virus on the head by boosting the T cell responses. But as I've already, hopefully I've got the point across. If you can get the T cells there in advance, then obviously that's really good. So this is the study, and this is just, I'll give you a little bit of detail for those of you who are interested. We've broken the virus up into small pools, which are represented by peptides or by whole proteins. And we're using these in the test tube. So you can take the blood from the vaccinee, offer them these small fragments of the virus, and then you can identify your specific T cells. And this is really what we found, and we're very excited by these results. So this is the lowest dose, that's a medium dose. But if you get up to a full dose, you can get large numbers of, in this case we're measuring the cells on a plate, a so-called ELISpot plate. So each individual cell each individual spot is an antigen specific cell, just exactly the same way as I showed you on those slides earlier. And so when we prime these individuals, we can produce spots which are in the thousands per million. So the actual numbers probably don't translate very well, but that's a very big immune response. It's bigger than what you'd normally see in a regular hepatitis C infection, and it's one of the strongest ones that's been generated by this method. So we're certainly able to introduce very big immune responses in these healthy donors. But we're not at the stage yet where we can know for sure whether these things are really protective or not, and that's the next stage, which is certainly going to be challenging for us. Now, one of the things that is important for us is that you don't just generate one immune response, which would be equivalent to one of those colours of squads. You need to generate multiple ones. So ideally, you'd have a set of different immune responses against the virus, which are all ready and acting at the same time, so the virus has got much less room for manoeuvre. And one of the exciting things about this regimen is because it's very immunogenic, some people are generating responses against all the proteins in the virus. So we've got a reasonable chance of pinning it down. It would have to make 6 separate mutations at the same time in order to get around this. So that's increasingly unlikely. And that same process that same sort of ethos is what's used in drug treatment for HIV, where you use multiple drugs to pin the virus down because it can escape. And this is just another picture. I thought you just might enjoy this. So this is very recent data from Ellie Barnes' trial, and we're using all the tools that the BRC Core Lab have helped us establish. And each of these dots is an antigen-specific cell. So we're able to boost, you can see about 5% of your immune response, if you count up, or 5 or 6%, the cells which are making these cytokines. And interferon gamma is a very potent antiviral cytokine, and TNF is another one which has got a role in the liver. And between them, the cells are making multiple cytokines. So these cells are actually making both, and there are reasonable numbers of them after treatment with these vaccines. So we're not only are the cells present and detectable, but we know exactly what they make, and these are the kind of things that we really would like them to make in order to be effective. But as I said, the proof will really be in future trials. And this is the last data slide, but these are using those tetramers again. And if you remember what we saw with the CMV, we saw a nice little cloud. And this isn't quite as big as with CMV, but it's a very decent, respectable one. And this is long after the initial vaccination. So we hope that we've got memory here that will last for a long period of time, so that if the person saw hepatitis C many years after the vaccination, they would still have those troops ready to fight off the infection. But Hep C is a tricky opponent, so it's very unpredictable. You never know what it's going to do. But I think at the moment, just to summarise the vaccine stuff, we've got the magnitude is good, so it's really easy to detect. My personal feeling is the breadth is good, and that's probably the main thing that we have to go for, have as many different things lined up. And what's also important to remind you is that, in fact, a fraction of patients with hepatitis C, unlike HIV, have natural control of the virus. So they'll get infected, and they will contain the virus, and a lot of that is driven by T cells. So we're not trying to invent anything new here. The body can already do it. What we're trying to do is encourage people to do it by boosting their T cells with a vaccine. So we're reasonably optimistic that we can make progress here, but there's still plenty of hurdles to overcome. So just to finish off, just final few thoughts. So host virus relationships are a bit like human relationships. So sometimes there's lots of activity at the beginning, and then nothing long term. And from the point of view of studying the immune system, that's not necessarily terribly exciting. That those are the things that our body can deal with anyway, and we perhaps don't really need to interfere. Sometimes it's perfect harmony forever. And that's also quite boring, to be honest, because those viruses don't necessarily cause a lot of disease. And we're actually full of viruses, and we keep discovering them, that are relatively harmless. And somehow their interaction with the immune system means that even if there's virus present, it's not causing a lot of pathology, and the immune system doesn't have to work extremely hard in order to contain it. So there's a nice relationship there. But from a clinical point of view, those are less interesting. The ones that cause us the most excitement are the ones where there are battles going on, and sometimes it's these you can't live with them and you can't live without them kind of battles. You can't get rid of the virus, but you can't contain the virus. Doesn't overwhelm the immune response. It's just a chronic infection with inflammation, and that may be the cause of many things beyond the diseases we talked about today. So in the course of the battles, humans have left their footprints on viruses, so you can see the impact of the immune response on the viruses and the way they've evolved. And viruses have certainly left their footprints on us, and you can see that in the way the immune responses have developed. And so it's a process which is ongoing, and we can even measure this process of coevolution within individuals and within populations as a whole. So I'd like to thank everybody. My salary up for the last period has been paid by the Wellcome Trust. My colleagues at the Peter Medawar building, particularly Ellie Barnes, and Gillian Griffiths, who produced the film of that T-cell killing the target, and the Biomedical Research Centre for organising the evening. So thank you very much. You can put the lights on.
I've got a question.
Yes.
You talked about is it 15% of people that clear the virus?
Yes.
Has anyone looked at what is different about